protein design problem at the beginning really seemed like kind of a lunatic fringe type problem with the idea the only proteins we knew were the proteins that came through natural evolution, and the idea that you could make new proteins really seemed kind of crazy. And the progress that has happened from the beginning is really the work of a whole community of many people who've worked really hard to make this happen and made contributions, at different stage in the development of protein design. I think there are, while this progress is really the work of a huge group of community of people, I think there are three contributions that, in my mind, really stand out. And these are the following. In 1988, Bill DeGrado, and Bill's here, and Lynn Reagan showed that you could write down sequences that were not biological sequences, that would fold up into, that would adopt folded structures. And that showed that you didn't have to go through evolution to come up with a sequence that could fold. And then in 1997, Steve Mayo and his colleagues showed that you could design proteins on the computer. You could take a protein structure and come up with a completely new amino acid sequence that would adopt that structure. And then a few years later, Brian Kuhlman, being familiar with the work of Bill and Steve, and knowing that my group was working on backbone sampling methods, had the idea of trying to do flexible backbone protein design, designing sequences to adopt new structures. And he then proceeded to do just that in 2003. And I really wanted to have this webinar so that everyone could understand how the field began, and and then how it's proceeded since then. And I'm really grateful to Bill, Steve, and Brian for generously agreeing to participate in this webinar. And I hope you all enjoy it. I think the way we're going to do questions, since we have a lot of people and not so much time, is just type them into the chat, and we'll choose some questions at the end after all three speakers have finished. So thanks everybody for joining. Thanks again, Bill, Steve, and Brian, and the show is yours, starting with Bill.
Okay, David, thank you very much for the opportunity to tell you a little bit about what we've been doing for the last 30 plus years. I first became really interested in proteins when I saw lysozyme structure as a undergraduate. I thought someday I want to make enzymes, and we're pretty much getting there. We started with a minimalist approach because simplicity is the ultimate sophistication. And we followed Einstein's dictum to make things as simple as they can be, but not simpler. I think we've illuminated a number of principles underlying structure and function, and this has led to both therapeutics and catalysts. Now, we started initially looking at self-assembly of peptides to protein-like structures, and this was a collaboration with David Eisenberg, shown here as a youth, and these are models that Linus Pauling used in the day to model things like helices, and those were precisely the same models that we used to design a 4-helix bundle. I was really glad, though, when we could transfer the coordinates to a computer and use energy minimization and state-of-the-art graphics to facilitate the process. Now, the sequence that came out of this is shown here with hydrophobic residues, all leucine, and charged residues plus and minus to electrostatically stabilize the bundle and give solubility. The peptide indeed formed a 4-helix bundle. But what exactly was the structure? AlphaFold 3 and Chi1 now allow us to go back and see what the structures of our original designs are. And gratifyingly, it looks just like what our design was. after this, we were able to include loops to make a dimeric and then a single chain for helix bundle, this being with Siuho and Reagan. Now, the protein, according to Chai-1, does fold into about the right structure, but it had low confidence. And at the time, we thought it was not as uniquely folded as a real protein, which I'm always delighted when things don't turn out quite right, because that's where the learning begins. Now, if you think about how a real protein folds, it starts in a continuum of states, goes into an equilibrium of intermediates, and then finally a native state wins. With alpha-4, what happened was we went into a very deep minimum, but not a unique one, not uniquely folded. And so the question is, how do we introduce conformational specificity? Well, one way is to build a zinc binding site. And so we did precisely that. And now quite nice to see that this is a high confidence model of the protein. And so indeed, already we had the first de novo protein with a native fold. We had the first switch, because zinc binding controls both the topology and the dynamics, and it's very similar to regulatory proteins today, like Caltech and a lot of things that are being de novo designed. Now, the next step, though, was also to think about the core packing. We had all leucine residues, all the same size, but proteins tend to have a geometrically complementary assortment of small and large amino acids that fit together in a jigsaw puzzle-like manner. And to facilitate this, Michael Levitt and Ponder and Richards went to the concept of rotamers, and that's that amino acids can only have certain shapes. And now we can treat those shapes in computer algorithms. And Desjardins and Handel made a genetic algorithm that really allowed one to go through and essentially try all side chains and low energy conformations to see what fits together the best. And we'll hear, of course, from Steve Mayo about the total automation of this process. About the same time, we were seeing coiled coils being designed based on sterics. And we, with David Eisenberg, solved the structure of what was expected from work from Hodges to be a parallel coiled coil. And instead, it was an anti-parallel coiled coil. Again, the unexpected delighted me because now you can think about connecting these helices. Of course, you have to make the protein much shorter, so it's not a coiled coil, but a globular protein. And so we shortened the helices, we dialed in a unique topology, and we used Desjardins' program to design the interior. And here you can see the NMR structure we solved in '99 looked just like the protein model. So this was the first de novo globular protein whose sequence and tertiary structure were designed from scratch and whose 3D structure was determined experimentally at reasonable resolution. Alpha 3D has been used quite a bit directly to study protein folding. It also has been outfitted with metal binding sites and made into catalysts, particularly in Pecораro's lab. And the company Arcellx used this as a framework for CAR T therapy constructs. And you can see here the small size compared to other things that are being used, and its durable stability really facilitated the properties. They now have taken this into phase 3B clinical trials, so it's progressing quite well. So an analog of alpha 3D was the first de novo protein in humans. And I think it still is by far the most advanced therapeutic in clinical trials. That, of course, excludes vaccines. Now, we can also use this for the first example of binding peptides, natural peptides. So we made a helix loop helix peptide that binds to the calmodulin binding domain of calcineurin. We see a nice saturable isotherm for the target, but it's very specific. It doesn't bind to myosin light chain kinase's calmodulin binding domain. And now if we look at Chi-1 prediction, we see that it's precisely as designed. We see the same thing from AlphaFold 3. And so we have the first de novo design of receptors for natural peptides with high specificity, and the subsequent paper we showed with low nanomolar binding affinity. With David Eisenberg, we also looked at domain-swapped 3-helix bundles. I was skeptical when Giovanna Ghirlanda showed me this model because the N-termini of the helices were butting one another. I was worried this might have electrostatic implications. But when Nancy Ogahara solved the structure with David Eisenberg's lab, boom, there was an anion right between them. Now, we could also use this domain swapping and crystallographic symmetry we saw to introduce high order oligomers, this now being hexamer created by a 3-fold and orthogonal 2-fold axes. Ultimately, we could design full crystals. Here we have angstrom level resolution accuracy over this length scale of 1 millimeter. We know where every atom is in a crystal. We also design metalloproteins, and you can think of them as being, again, composed of pieces of peptide and in a retrostructural manner. Here's a rubridoxin, and here's a diiron protein. It's really quite complex. But deep within the active site, you see a reasonably simple 4-helix bundle, each helix being more or less equivalent with point group symmetry and having a single glutamic acid. So now we can go and look at a large number of structures of natural proteins. We can get structural embeddings by parameterization. And now we can use those embeddings to build up a full 4-helix bundle entirely mathematically. So every atom in here is defined by an equation. And what we see is an ideal structure, an excellent agreement between experiment and computation in this structure solved by Angela Lombardi's group in 2000. She also worked on the design when she was in our lab. So we have now the first high-resolution structure of a de novo protein. We also, over the years, were able to tune the reactivity for either 2 or 4 electron chemistry, getting very different products from the same substrate. We departed from helical bundles for a bit in making beta barrels here that bind to iron in a rubridoxin-like site. In work sort of thought of early by Dayhoff from gene duplication. We designed the first heme proteins. This is a multi-heme protein designed with Les Dutton, and much later, we started combining them with the diiron sites. And you can see now the diiron oxidase activity is allosterically regulated by the binding of a porphyrin. Now, we've had a long interest in membrane proteins. Let me just show you one thing that we've done. In early work, 88, we designed a peptide that formed tetramers and a different sequence that formed hexamers, this being a proton channel and this being an alkali metal channel. If we now look at what the structures are predicted to be using Chi-1, it's remarkable. This is a stunning agreement with what we had published in 88 using energy minimization. We can also go on and design transporters that exchange protons and zinc ions. So what did we accomplish in these missing years? We defined the hierarchy of interactions required for folding. For binding, we were already able to make switches, specific peptide binders, and peroxidase catalysis. Membrane proteins, we had both structure and function. We made alpha 3D, which you see is now quite advanced. And we had point group assembly, really designing large assemblies. So enough with the history, though. What are we doing now? So one thing that's still a challenge is designing proteins that are very specific for small molecule binding, especially polar molecules. With this, we can make reversal agents for toxins and drugs. We can make carriers to change pharmacokinetic properties. And that's exactly what we've done. Here is a protein that binds rubidoxin, designed entirely from scratch with 5 nanomolar KD or lower. If we can, in vivo, we can see if you dose rucaparib here, it decays or is cleared. But if we add the protein, it binds and it's renally cleared much more rapidly. Here is another example. This is a pixaban, and we have both the L form, but we can also chemically synthesize the D form. Now, the L form, when we look for its immunogenicity, it has some, but the all D form has none because it can't be broken down to peptides that are then expressed in or bound to MHC and then initiating an immune response. Nevertheless, the all D form was fully active. So can we design enzymes computationally? A benchmark that's used quite a bit in previous work in which people took computation to redesign the active site of natural enzymes, and they used this reaction to see how well they were doing. Well, could we do it completely by de novo? And could we do it better? And indeed we can. We were designed this bundle here to bind to the transition state analog. How do our first, right off the computer designs compare to previous work? And you can see we're about a factor of 50 to 100 better. Previous workers also used directed evolution to improve their activities. We did one round and said, okay, that's enough. It's 10 to the 5th per molar per second. Ultimately, one, yes, can reach that here after 17 rounds, or with really nicely improved directed evolution methods, do this in 5 rounds. But ultimately, enough with these model reactions, coumarin ester hydrolysis, nitrophenyl ester hydrolysis, Kemp elimination. We really need to make enzymes that do things that are useful. So we thought about what is everything we want in an enzyme. We'd like it to be able to take either a natural or artificial cofactors. It should be small and atom efficient. Substrate concentrations. We need it to work at molar concentration. We don't want to be wasteful of solvent and run at low concentration. It should be evolvable. We can't do everything perfectly the first time, and we'd like it to be evolvable in cells. Thermal stability is, of course, important. And what hasn't been looked at much is stability to organics. Now, Kai-Peng Liu in our lab has worked in collaboration with Yang Yang and Peng Liu to design an enzyme that catalyzes this non-natural reaction, cyclopropanation, that introduces 2 chiral centers. He docked a virtual transition state into the active site in an approach similar to what Hauck had introduced years ago with David. And what he finds is with 0.25 molar substrate, he gets 40,000 turnovers. Now, did he have to make a lot of things? No. 4 out of 10 of his designs showed more than 99% in anti-Americ ratio. This matches the most successfully engineered natural proteins. And he also showed that these are stable in ethanol. This is really important because you want a lot of your substrates aren't soluble in water. And this is the greenest of solvents. And so we can see we have still full activity at 70% ethanol. He went on and also did a silylation reaction where we're inserting a carbene between a silane and hydrogen. Again, a chiral center is created. Initially got 3,000 an anti-Americ ratio in 0.2 molar substrate. But ultimately, with a few rounds of directed evolution, was able to get to 30,000 turnovers. And that's 10 times the best engineered natural enzyme. It's stable up to 60% methanol in this case. And then finally, let me close by saying we're starting to design components for much larger proteins that have been designed via protein engineering. So here we were interested in how signals propagate from binding to a periplasmic domain to inside a cell in histidine kinases. There are a lot of hypotheses, but we could test them by making a zinc binding protein here and show that it's signaled in an appropriate way. We also can improve the properties of ion channels that are used in DNA sequencing, shown here. And this is Lee Schneider's work. Well, I want to thank all the workers for all these years. Most of you weren't part of the de novo protein design effort, but you all contributed to our atmosphere and our environment and come from many, many different countries and have returned to many. So thank you very much. And sorry for going over a little.
Well,
thank you, Bill.
Bill.
Okay, great. Can you hear me now? Okay, thank you. So I guess the first thing I want to do is thank David for this this webinar. I think it's a great idea. And obviously I want to congratulate him and his lab on winning the Nobel Prize in Chemistry, a great achievement for the field. So what I'd like to do is sort of focus my 15 minutes on talking about our work between 1997 and 2001. And I'll get right into it. Here's a sort of a brief outline. I'll provide some historical context from a personal perspective, kinds of things I was thinking about at the time, talk about the design approach and implementation, and then talk about methods optimization as an example of methods optimization and how that led to the design of hyperthermophilic protein variants. Then I'll talk about our full sequence design, talk about sort of an early example of computational design with backbone flexibility, and then talk about computationally designed enzyme-like protein, all very briefly. Okay, so I sort of came into the arena thinking about this problem with a background of having co-developed molecular design and simulation software that was being developed for mostly small molecule drug design. And also at the time, I was very involved in co-developing a generic molecular mechanics force field that would be usable for both proteins and small molecules. And that work was done with Barry Olesen and Bill Goddard, along with the company as well. So in the sort of late 80s, early 90s, as I was thinking about what I would do as an assistant professor, I was very aware of, impressed with the work that Bill DeGrado and coworkers were doing, as we just saw, really impressive stuff. I was really influenced by Carl Pabo's Nature letter, where he laid out the rationale for the inverse folding problem and argued that it was a way to think about protein design. That is, have a backbone first and then compute or design a sequence for that backbone. And by using sort of the inverse folding approach, you could avoid the problem of having to predict the structure of the protein. Of course, the side chain rotamers, as developed by Ponder and Richards, and then further by Dunbrack and Karplus, was very influential, as was the computational method, dead elimination, developed by Johann Desmay et al. And of course, at the time, we were very aware of the work that John Desjardins and Tracy Handel were doing on computational design of hydrophobic protein cores, which was also mentioned in Bill's talk. So our goal at the time was to solve the inverse folding problem by developing a fully automated computational approach that leveraged an iterative design cycle for methods optimization. Okay, and so what is the design approach? We have a fixed protein backbone, discrete sidechain rotamers, an atom-based force field, combinatorial optimization methods like dead-end elimination or Monte Carlo, a negative design to help enforce fold specificity. And then, of course, we want to be able to iterate our designs and learn from this, design, build, test, analyze cycle on how to modify our procedures so that we can actually get better designs. And then our approach, we were interested in segregating the protein into core, boundary, and surface regions. So the buried hydrophobic core, the fully exposed surface, and then the boundary between those regions. And then to develop methods that were optimized for each of these regions before sort of combining them into a single method or full sequence design. Okay, the basic implementation is shown through this little cartoon. So here's a simple problem. We're going to design two positions on this cartoon protein, positions P1 and P2. We need to select amino acid side chain rotamers. And the important consideration is that you're allowing rotamers from multiple types of amino acids. And that's how you get the design. And so that's an extension from what Desmay et al. Had done for rotamer optimization for single sequences. And then we use a two-body, we use an energy composition that includes both one-body terms and two-body terms. One-body terms are computing the interaction energy between an amino acid side chain rotamer and the rest of the backbone in the absence of other side chains. And then, of course, we do that for all the rotamers at all the design positions to fill out this vector. And we use a collection of potential functions that describes the interactions within the protein, sort of standard things that you might imagine: van der Waals potential, electrostatics, hydrogen bond potential, and a solvation potential. Then we move on to the two-body interactions. In that case, the backbone goes away, and we're now looking at all pairs of rotamers to fill out this matrix for two-body interactions. And so then with the energies computed, we have a combinatorial optimization problem that we need to solve. Simplest thing to do is use Monte Carlo. And so in this simple example, we can get the ground state solution pretty easily because it's very small. And we see what the solution is mapped back onto our structure. And we spent a lot of time in the early days thinking about optimization algorithms. We ultimately landed on using dead-end elimination because of its ability to achieve ground state solution. And we believe that that was important in the context of trying to optimize methodology in order to be sure that your method optimizations are going to not to be applied to sort of the noise that you get from a stochastic optimization algorithm. But we were very engaged in, Monte Carlo, genetic algorithms, self-consistent mean field approaches, and FASTER, which is a combination of several techniques together. Okay, and so as an example of method optimization, we were interested in tuning the van der Waals potential in order to accommodate for the fixed protein backbone and discrete sidechain rotamers. And so one of the problems in the early design attempts was that because of the discretization of the sidechain rotamers, you could get elimination of lots of things because of steric clashes due to the van der Waals potential. And so we did a simple thing, which was to define a scale factor alpha on the van der Waals radii that we plugged into our Leonard-Jones 612 potential. and then just varied the scale factor and looked at various design results for designing the core of protein G. And then you can see here that, as you decrease the scale factor, the relative core volume relative to wild type protein G increases. If you have scale factor above 1, the relative core volume decreases. And so then we made all these molecules and just assessed which one gave the best result. And so in this case, using a scale factor 0.9 yielded a protein that had a far UV CD spectrum indistinguishable from wild type protein G, and a thermal unfolding curve that yielded actually a more stable protein. Then by NMR, you can see 1D NMR, 1D protein NMR, you can see that the dispersion in the fingerprint region is as you would desire. But then if you go down to just alpha 0.85, you end up with a protein that is with significant line broadening, suggestive of potentially a molten globule. And then if you look at the modeled structures, what you see is that in the alpha 0.85 case, we get an exposed hydrophobic residue, which probably leads to some level of protein aggregation. So we settled in terms of optimized alpha value of using 0.9. And then if we apply that to a larger design case in protein G still, where we're looking at all the core and boundary positions, what we see is that we can design variants that are hyperthermostable. And this is what you would expect if you have an optimized potential function and you're using a combinatorial optimization method that actually gives you ground state solutions. And so just as one example, here's the fully designed variant C3b4. It is does not unfold at 99 degrees C compared to wild type protein G. If we do chemical denaturation at 50 degrees C, we can see that the design protein unfolds with over 4 kcals per mole of additional free energy of stabilization. Then we can solve that structure by NMR, and it's indistinguishable from the wild type protein G structure. So we did development like that for the various regions of the protein and then combined them together to have a unified methodology for computing sequences for an entire protein. And in order to test that, we turned to the zinc finger fold ZIF268, which is you can see as the helix, a very small beta sheet and a turn stabilized by the binding of a zinc atom. And so in our hands, what we wanted to do was to remove the requirement for zinc binding and compute a sequence that would actually yield this fold. And so that calculation was combinatorial complexity of 10 to the 27 amino acid variants. We can, here's the sequence, full sequence design 1 compared to ZIF268. You can see the sequence is very different. If we, for example, it's a very novel sequence. If we blast this sequence against the sequence database, we get hits that have, as high as 36% sequence identity, but the p-values for those hits are statistically insignificant. They range from like 0.6 to 1.0, so highly statistically insignificant. If we take generate random sequences from the same design composition that we use, amino acid composition that we use for the design calculation, we get very similar hits from BLAST with sequence identities around 36% and p-values that range from around 0.3 to 1.0. So the FSD1 sequence is truly a novel sequence. And as we all probably know, it folds the correct structure, and compared to its design target, we get a good overlap. So we believe that this was the first real demonstration of a solution to the inverse folding problem, and really set the methodology for additional advancement in the future. Okay, then very briefly, I just want to mention something about backbone flexibility and sequence design. So in 97, Alice Su in the lab implemented an approach to look at coupling backbone flexibility to sequence design. And in her hands, what she did was to define sort of supramolecular parameters that define the structure. And so a plane for the beta strand, a cylinder for the helix, and then we would vary these parameters that control the orientation of the helix with the beta sheet plane, including looking at distance from between the two elements. And so a series of changes in the distance between the helix and the sheet resulted in various designs, all of which are folded and stable and give NMR spectra that have dispersion that you would expect for a fully folded protein. And the one point I really want to emphasize here is that for flexible design, the flexibility and the sequence design are not fully integrated. So you do flexible design, and then with that new fixed backbone, you do sequence design. And so there's this, I think, tight reliance on having to go back to do a fixed backbone sequence design, even if you're doing flexible design. And then very briefly, I wanted to just show an example of what we think is probably the first example of a computationally designed enzyme-like protein. This is for ester hydrolysis of this substrate. It goes through some high energy state and then yields product. The way we implemented this was by just creating rotamers of the high energy state and then inserting that into the protein and looking for design mutations in the protein scaffold that actually stabilize the energy of this state. That works. You can get an active molecule that shows saturation kinetics, burst phase kinetics, and other features that are hallmarks of naturally occurring enzymes. And with that, I would like to just acknowledge more directly the people who did the work that I talked about today. Basil Dahiat was instrumental in the full sequence design work. Sandy Malakauskas did the hyperthermostable protein design work. Alice Su did the flexible backbone work. And Dan Bolin did the enzyme-like protein work. And I'll stop there. Thank you. There.
Great. Can everyone hear me? Thanks, Bill and Steve, for wonderful talks. And thanks, David, for the invitation to speak today. I'm going to keep my talk pretty focused. So in the first part, I'm going to revisit some of the methods that we used to design Top 7. And then I'm going to segue into some of the things that I think we learned from the Top 7 project and how that relates to current strategies that are being used for protein design. So when I joined David's lab as a postdoctoral fellow, Steve's lab had already shown that computational methods could be used to design new sequences for existing protein backbones. Bill's lab had shown that you could design a variety of different helical bundle proteins. And also Per Harbury and Peter Kim had shown that they could use molecular modeling to create new coiled coil structures. And so we were interested in pushing a little bit further in designing a mixed alpha-beta protein with a fold that at that time had not been seen before in the PDB. And so we picked this as our target fold. It includes 5 beta strands and 2 alpha helices. And so the problem that we needed to solve was how are we going to go from this low resolution picture of what we wanted to design to a high resolution model and a sequence that we could make and validate in the lab. And so we divided this up into two primary steps. One being backbone generation, so creating protein backbones that adopt our target topology. And then in the second step, sequence design, so the inverse protein folding problem that Steve mentioned. And so I'm just going to dive into each of these a little bit more. So first, for the backbone generation, we were able to make use of some advances that were happening in protein structure prediction in David's lab, and particularly the development of the molecular modeling program Rosetta. And we settled on a protocol that could best be called fragment assembly with constraints. So this had three basic steps. So first, assign the desired secondary structure for each residue in the protein, and then pick small pieces of naturally occurring proteins with those desired secondary structural elements. And then piece those together with Monte Carlo sampling with a score function that includes constraints that will specify your target fold. So for instance, a constraint between these two residues would dictate that you want them to be near each other in the final folded structure. And so the set of constraints that we used to design top 7 are here. And these were basically constraints between residues that we wanted to be hydrogen bonding with each other in the beta sheet that we were creating. And so with these constraints and this target secondary structure, we were able to build backbones that would adopt our target fold. And some of the, I think some of the advantages of this approach were that by building these structures from protein fragments, we were ensuring that the local structure should be designable. Secondly, Rosetta had already been trained to build native-like beta sheets. So the beta sheet should be designable as well. And then thirdly, one nice feature about this is that it's a stochastic protocol. So we could use independent trajectories to produce different backbones. And this could become important if some backbones turned out to be more designable than others. So then with our backbones, we then needed to design sequences for them. And we were heavily inspired here by work from the Mayo lab, as well as earlier work by Ponder and Richards to use a rotamer-based approach to solve this problem. So here again, the problem is you have a target protein structure that you'd like to create. You need to find an amino acid sequence that will stabilize the structure. So form good hydrophobic packing, good hydrogen bonds, and good electrostatic interactions. And we settled on a pretty simple algorithm for solving this problem. One mentioned by Steve, which was just to use Metropolis Monte Carlo. So we would just start with a random sequence and then make single substitutions and evaluate those changes with the Metropolis criterion, using an energy function that favored the terms that we know are important for protein stability, including van der Waals forces, hydrogen bonding, an implicit solvation model, and torsion energies. And some of the things that I think were important about this protocol is that the way that we created it, it was relatively fast. So you could design a new sequence for a whole protein in just a few minutes on a single CPU. And so what this allowed us to do is we could run it on many different protein backbones. And that will become important when I get to the next slide. But another reason why that was important is what it meant we could do is we could design new sequences for large sets of naturally occurring proteins. And what this allowed us to do is train our energy function, set the weights in our energy function to reproduce native amino acid sequences. And so in this way, we could implicitly design for both stability and solubility with a single protocol. The other thing that's pretty interesting about this is actually the approach that's being used in the modern day to train neural networks for protein design. So models like protein MPNN are basically being trained to reproduce native amino acid sequences. So with these two things in hand, we were able to design a protein models that look pretty good by eye, but when you went and scored them with your energy function, you saw that they had energies that were significantly worse than naturally occurring proteins. And our hypothesis for what was going on there is sort of illustrated in this simple cartoon, where you create an initial backbone that looks pretty good, but when you try and design a sequence on it, some of the contacts really aren't that optimal. And so what this led us to do is then think, well, then maybe we need to refine the protein backbone. So if you refine the protein backbone, maybe you can make some of the contacts better. But now that you've changed the backbone, the ideal sequence for that backbone may be different. And so this suggests that you should do some more sequence design. And then when you do that, for instance, maybe the best two amino acids here changes. But now, of course, you have a new sequence, which means the preferred backbone may have changed. And so this suggested to us that we should really use a protocol that iterated back and forth between these two steps, doing sequence optimization followed by structure refinement. And so this is one of the reasons why it was really important that we had a fast protocol for doing this sequence design. And so the final protocol that we used for designing top7 involved building the starting backbones, as I described, and then coupling sequence design with structure refinement. And what was nice is this protocol was fast enough that we could run many independent trajectories and create thousands of models, and then sort them based on calculated energies to pick out our favorite ones to characterize in the lab. And so we went through 2 generations of designs characterizing 2 or 3 sequences in each case. And in the second generation, we were lucky that one of the proteins we were able to crystallize. And what my collaborator Gautam Dantas found is that the crystal structure of our design sequence closely matched the design model. And so this was true both at the backbone level as well at the side chain level. And this was performed in the lab of Barry Stoddard. And so this was really exciting result. And I think some of the things that we learned from the top7 project, which sort of build on the things that Steve and Bill have already been telling us about, is it further highlighted that it's possible to use computers to create new protein structures. And we did this by focusing on hydrophobic burial, side chain packing, satisfying hydrogen bonds, and using idealized connections between secondary structural elements. The other thing that we saw, and this is similar to one of the results that Steve showed, is that top7 was incredibly stable. So when you heated it up to 100 degrees Celsius, it remained completely folded. And we had to add up to 5 molar guanidine to get the protein to unfold. And this is interesting because if you compare this to naturally occurring proteins, they're generally not this stable. And so this tells you something about protein evolution. Evolution could make proteins that are this stable because it is physically possible, but it hasn't seen the reason to do so. A couple of things that I think were important for success in this project was starting from a designable backbone, and then also using the iterative structure prediction design to find lower energy structure sequence pairs. And I think both of these points have been key factors in the many protein design projects over the years. And so I just wanted to highlight that even now with the modern AI-based methods, it can be very useful to sort of engage in this iterative process of structure prediction and sequence design. So here I'll just share one result from my lab using a pipeline that we call EvoPro that uses a genetic algorithm to optimize a sequence pool for a given fitness function. And so starting from a pool of sequences, you predict their structures with AlphaFold. You then rank those sequences using either confidence scores or other features that you want the design proteins to have. You throw away the poor performing sequences and you refill the sequences by either using AI-based methods for protein design, such as protein MPNN, or introducing random mutations. And so in this particular pool, we just wanted to take some small de novo design proteins from the Baker lab that were really stable, but didn't inherently bind anything, and mutate their surfaces so that they could bind to a target protein. And so when you go through this iterative optimization of sequences and structures, you can see that over 50 generations, you start to converge on sequences that are predicted to adopt the target complex with really high confidence. And then you only have to take a small number of these into the lab. So in this case, we characterized on the order of 20 of them, and more than half of them showed evidence for binding. And some of them bound quite tightly straight from the computer. So here's one example right here that bound with a KD of 1 nanomolar. Just really highlighting the power of these new AI-based methods, something that would have been really hard to do, I think, with the tools that we had available 10 years ago. The other thing that was, I think, really important in the design of Top 7 and many other protein design projects is it's really important to start with a designable backbone. And so over the years, a variety of methods have been explored for doing this. This includes some of the work by Per Harbury and Peter Kim, where they used the parameterized representation of the coiled coil to sample different coiled coil backbones to search for ones that were designable. I told you about in the Top 7 project about using local structural fragments to ensure designability. People have developed methods that actually use tertiary fragments from proteins, or other methods that dock proteins together in a way where the interface should be inherently designable from that docked position. And then most recently, we've had these really powerful AI-based methods that have been developed, such as RF diffusion, which can create protein backbones that are significantly larger than we could do with the previous methods. And what's been really exciting is that many of these backbones are designable right from the beginning. You don't even need to do any further structure refinement to come up with sequences that will fold into those structures. And so I'd just like to finish with, I think, just a summary of where I think we are in the field right now, what we're really good at, and some of the things that we'd like to get better at over the next few years. So I think right now, especially with the new AI-based methods, we have robust pipelines for designing highly stable, well-ordered proteins and complexes. And so this is useful for creating protein inhibitors, presenting epitopes for vaccine design, and stabilizing naturally occurring proteins. Moving forward, these are problems that we're already working on, but I think we still need to get better at, which is the incorporation of flexibility and allosteric inner designs and functional loops to enable the design of more diverse functions. And so the applications for these types of features will be de novo enzyme design, de novo antibody design, switches, and even more ambitiously molecular machines. And so just to conclude, I'd like to acknowledge the people that worked on the particular projects that I talked about, but I'd also like to really give a big shout out to the Rosetta Commons community, which has been a wonderful community to be working in. And then I'd also like to give a shout out to all the people in the protein design field that have made their tools accessible to all of us. And this has really been critical to the success of our field. So thank you.
Oh, well, thanks, Bill, Steve, and Brian, for really outstanding talks. And I think we have time for a few questions. Ian?
Yes, we do. Hi, I'm going to read off some of the top questions that have come in. We have more questions than time, so I'll sort of run through these. So I'm combining two of the top questions here. They both have to do with the dynamical aspects of protein structure, and this one I'm going to throw to Steve. So the two questions are, how would we incorporate dynamics into models such as AlphaFold, and are methods such as molecular dynamics still useful in the age of AI?
So yeah, dynamics is obviously critical. I'm not sure if I can answer the question with any level of precision in a short period of time. So one way to think about modeling dynamics in design is to use an ensemble of backbone structures that represent the sort of the potential dynamical motion of the protein, and then to do a sequence design across the entire ensemble where you're constraining the sequence to be the same on every member of the ensemble. So we actually worked on developing methods like that. to do sort of coupled design, but never at the level of fully exploring the dynamical motion of a protein. And then what was the second part of the question about AlphaFold? Sorry.
Could we incorporate dynamics into models like AlphaFold?
Um, I'm sure the answer is not to say no, but I don't know how to do it proactively. Yeah, so almost certainly yes, but I'm not sure how you would get there.
Very fair. All right, another question. I'll start it with David and then whoever wants to take it. AI has undoubtedly revolutionized our ability to predict and design protein structures, but we haven't necessarily solved the problem from a first principles approach. Do you think we will ever achieve this, or are AI-based methods getting so good that focusing on first principles may no longer be necessary?
I think it comes down to what one means by first principles. The force fields that Steve and Brian described have many parameters that were determined in quite a range of ways, and the deep learning models obviously have way more parameters. I suppose someone might say that a first principles approach might be obtaining all the parameters of a model from a high-level quantum chemistry calculation, but that's hardly feasible in the context of a protein and solvent. So so I think one has certainly gained a lot of insight into protein folding from all the design work that's been done, as I think all three speakers highlighted. The fact that design proteins can be extremely stable suggests, as Brian said, that evolution has not really sought to maximize protein stability. One of the things that we found really early on is that design proteins fold very fast. They fold no slower than naturally occurring proteins, sometimes faster, suggesting that evolution has not optimized protein folding rates or pathways. So I think that question is a little bit open-ended. I don't any of you would like to build on that. I'll just say that Bill's approach, was Bill was designing directly from knowledge of physical chemistry. There were very few parameters in his design work, so that may be the closest.
All right, and a forward-looking question to end with. I'll modify it a little bit. For someone who's thinking about getting into the field of protein design, what's the best way to do that today? Bill, Brian, and anyone.
Good question. I'd say find a lab that does protein design if you can. Take as many tutorials if you can't, and apply these methods of AI make it just so much easier now than in the old days. So that I think it becomes feasible to really design proteins.
Yeah, I think I would just add that I think the real exciting thing now is all the applications that we can apply protein design to. So also be on the lookout for problems, interesting problems in labs where you could maybe bring two labs together, a protein design lab with another lab with expertise in a certain field. And that's a great way to work on really impactful problems.
Yeah, I would echo that. I think the real future for protein design is really in applications, and looking for applications in either medicine or environmental remediation, things like that. I think are great ways to get into protein design.
Terrific. And with that, we are probably at time. So David, would you like to wrap up?
Yeah, I'd just like to thank Bill, Steve, and Brian again for really outstanding presentations. I asked them if they could sort of recreate sort of what was going on in their minds at the time of these fundamental discoveries and what happened. I think we got really a unique opportunity to hear. So I tremendously I learned a lot and enjoyed it. I hope all of you did as well. And as I think everyone said, I think there's huge potential moving forward, and hopefully this helps inspire a next generation of scientists to to make the next advances in this field. So thank you all for joining.